A vaccine is, well, a tool. It's a biological tool designed to teach our bodies how to fight a specific enemy. That enemy is a germ, like a virus or a bacterium. The goal here is to create immunity without causing the actual disease. For a long time, the definition of a vaccine was very simple. It was a preparation made from a germ. This germ was either killed or weakened, so it could not cause serious illness. Our immune system would see this safe version of the germ. It would learn to recognize it and build defenses like soldiers preparing for a real battle. This way, if the real dangerous germ ever invaded, our body would be ready to defeat it quickly and effectively. This classic idea of a vaccine has served humanity well for centuries. It gave us powerful weapons against diseases like smallpox, polio, and measles. Scientists would grow large amounts of a virus or bacteria. Then, they would use chemicals or heat to inactivate it, making it harmless. Or, they would pass the germ through animal cells over and over again until it evolved into a weaker form. This weakened or attenuated version could still replicate a little in the body, which was good for creating a strong immune response, but it would not cause the full-blown disease. This was the bedrock of vaccinology, a reliable method for training the immune system. However, science never stands still. Our understanding of biology and genetics has grown immensely. This has led to a big change in how we think about vaccines. We now have new kinds of vaccines that do not use the whole germ at all. Instead, they use a small piece of the germ's genetic code. These are called nucleic acid vaccines, like mRNA and DNA vaccines. They work by giving our cells a set of instructions. These instructions tell our cells to build just one piece of the virus, usually a protein from its surface. Our immune system sees this protein, recognizes it as foreign, and builds its defenses against it. This new technology has expanded the very definition of a vaccine. It is no longer just about using a weakened or killed germ. It is now about delivering specific information to our immune system in the safest and most effective way possible. This shift from using the whole pathogen to using a tiny genetic blueprint represents a major leap forward. It allows for faster development and potentially safer vaccines, as we are not introducing the entire virus or bacterium into the body. The fundamental goal remains the same, to create immunity, but the methods we use to achieve that goal have become much more precise and sophisticated. In 1955, the United States was on the verge of a historic victory. Dr. Jonas Salk had developed a vaccine to fight polio, a terrifying disease that paralyzed thousands of children every year. The Salk vaccine used an inactivated or killed polio virus. The idea was to show the immune system the virus without any risk of it causing the disease. After successful and extensive clinical trials, a massive national vaccination campaign began. Parents lined up with their children, full of hope that the nightmare of polio was finally ending. It was a moment of great national pride and scientific achievement, a symbol of progress against a dreaded enemy. But this moment of triumph quickly turned into a tragedy. One of the companies licensed to produce the vaccine was Cutter Laboratories in California. In their rush to manufacture millions of doses, a catastrophic error occurred. The process for killing the polio virus was not done correctly. Some batches of the Cutter vaccine contained live, fully dangerous polio virus that had not been inactivated. Unknowingly, doctors and nurses injected more than 120,000 children with a vaccine that was supposed to protect them, but instead gave them the very disease it was meant to prevent. This was a failure of manufacturing, a breakdown in quality control. The results of this mistake were devastating. About 40,000 children who received the faulty vaccine developed a mild form of polio. Another 200 were left with permanent paralysis. Tragically, 10 children died. The incident, which became known as the Cutter incident, sent a shockwave of fear and distrust through the public. The entire polio vaccination program was temporarily halted as officials scrambled to understand what went wrong. It was a dark day for public health and a stark reminder that even with a brilliant scientific discovery, the execution must be flawless. The process of making a vaccine is just as important as the science behind it. The Cutter incident became a pivotal moment in the history of vaccine regulation. It exposed serious weaknesses in the government's oversight of vaccine manufacturing. In response, federal authorities created a much stricter system for testing and approving vaccines. They established new standards for production and implemented rigorous batch testing to ensure that every vial was safe and effective. This terrible failure led directly to the strong regulatory framework we have today, which is designed to prevent such a disaster from ever happening again. It was a painful lesson, but one that ultimately made all future vaccines in the United States much safer for everyone. In the 1960s, scientists set their sights on a common but serious childhood illness, respiratory syncytial virus, or RSV. This virus is a major cause of pneumonia and bronchiolitis in infants and young children. Researchers decided to create a vaccine using the traditional method of the time. They took the RSV virus, grew it in a lab, and then treated it with a chemical called formalin. This process was meant to inactivate or kill the virus. This formalin-inactivated vaccine was then tested in clinical trials involving infants and toddlers, the very population most vulnerable to severe RSV disease. The scientific community was hopeful that they could conquer another childhood scourge. The trial began with high hopes, but the outcome was the opposite of what everyone expected. Not only did the vaccine fail to protect the children, but it also made the subsequent natural infection much worse. When children who had received the vaccine were later exposed to the RSV virus circulating in the community, they became far sicker than the children who had not been vaccinated. They developed unusually severe pneumonia and high fevers, a phenomenon that scientists had never seen before. This unexpected and dangerous outcome was alarming, and the trial was immediately stopped. It was a clear and frightening failure. The tragedy deepened as the consequences became clear. Many of the vaccinated children had to be hospitalized, and 2 young toddlers in the study died from the severe disease. Scientists were faced with a puzzle. Why did a vaccine designed to protect against a virus make the illness more severe? The investigation that followed revealed a new and complex biological problem. The formalin-inactivated vaccine had triggered the wrong kind of immune response. It stimulated the body to produce antibodies, but these antibodies were not effective at neutralizing the virus. Instead, they formed complexes with the virus that clogged the small airways of the lungs, causing massive inflammation. This phenomenon became known as vaccine-associated enhanced respiratory disease, or VAERD. It was a profound and humbling lesson for vaccine developers. It taught them that the immune system is incredibly complex, and that simply showing it a piece of a killed virus is not always enough. The way a vaccine is prepared can dramatically change how the body responds. The failure of the 1960s RSV vaccine set back research for decades, but it also forced scientists to dig deeper into immunology, leading to a much more sophisticated understanding of how to design vaccines that generate a safe and protective response, a lesson that is still critically important today. In early 1976, an outbreak of a new influenza virus occurred among soldiers at Fort Dix, New Jersey. The virus was identified as a swine flu, similar to the one that caused the devastating 1918 flu pandemic. Public health officials became deeply concerned. They feared that this could be the start of a major global pandemic. The memory of 1918, which killed millions of people worldwide, was still a powerful force in the minds of scientists and policymakers. Acting on the advice of leading experts, President Gerald Ford announced a bold and ambitious plan to vaccinate every single person in the United States against this new swine flu. The government launched a massive, fast-tracked program to develop and produce a vaccine. Pharmaceutical companies worked around the clock to create millions of doses. The goal was to get ahead of the virus, to build a wall of immunity across the nation before the flu season began in the fall. The campaign was promoted with the slogan, "Get a shot of protection." It was a huge logistical undertaking, a race against time to prevent a potential catastrophe. The public was urged to participate, and mass vaccination clinics were set up in schools, community centers, and workplaces across the country. However, the feared pandemic never materialized. The swine flu virus that appeared at Fort Dix did not spread beyond the initial cluster of cases. It seemed to fizzle out on its own. Meanwhile, the vaccination program was well underway, and millions of Americans were rolling up their sleeves. As more and more people received the shot, reports began to surface of a rare but serious side effect. Some people who got the vaccine developed a neurological disorder called Guillain-Barré syndrome, or GBS. This condition causes the immune system to attack the nerves, leading to muscle weakness and sometimes paralysis. The link between the vaccine and GBS, though small, became a major public relations disaster. The risk was estimated to be about 1 extra case of GBS for every 100,000 people vaccinated. But the story of a vaccine causing paralysis was terrifying to the public. The program, which had started with such urgency and good intentions, was now seen as a dangerous overreaction. The government was accused of scaring people into getting a vaccine for a pandemic that never happened, only to expose them to a harmful side effect. In December 1976, the National Swine Flu Vaccination Program was officially suspended, a clear admission of failure. Rotavirus is, a common and highly contagious virus that causes severe diarrhea, vomiting, and dehydration in infants and young children. Before a vaccine was available, it was actually the leading cause of hospitalization for this reason in the United States and a major killer of children in developing countries. For years, scientists worked to create a vaccine to prevent this serious illness. In 1998, their efforts finally paid off with the approval of RotaShield. It was the first vaccine licensed to protect against rotavirus. The medical community really celebrated this breakthrough, believing it would save countless young lives and prevent immense suffering. RotaShield was an oral vaccine given as drops in a baby's mouth. It used a live, weakened form of the virus to stimulate a strong immune response in the gut, where the rotavirus infection actually occurs. The initial clinical trials had been very successful, showing that the vaccine was highly effective at preventing severe rotavirus disease. Following its approval by the Food and Drug Administration, the Centers for Disease Control and Prevention recommended that all infants in the United States receive it. Doctors began administering the vaccine to babies across the country, hopeful that they had a powerful new tool in their arsenal. But soon after the vaccine was widely introduced, a worrying pattern began to emerge. The CDC started receiving reports of a rare but very serious bowel obstruction called intussusception. This is a condition where one part of the intestine slides into an adjacent part, kind of like a telescope collapsing. It is a medical emergency that can be fatal if not treated quickly. The reports suggested that this was happening in some infants within a week or two of them receiving their first dose of RotaShield. Public health officials immediately launched an investigation to determine if the vaccine was responsible for this problem. The investigation confirmed their fears. The data showed a clear, though small, increase in the risk of intussusception among infants who had received RotaShield. The risk was estimated to be about one additional case for every 10,000 babies vaccinated. While this was a rare event, the condition was so severe that the risk was deemed unacceptable. In 1999, less than a year after it was introduced, the manufacturer voluntarily withdrew RotaShield from the market. The vaccine that had held so much promise was now considered a failure, not because it didn't work, but because its safety profile just wasn't good enough for a universal infant vaccine. Section 6. Challenges with bird flu. The problem of a moving target. Influenza viruses are masters of change. They are constantly mutating, creating new strains that our immune systems have never seen before. This is why we need a new flu shot every year. The vaccine must be updated to match the specific strains that are expected to circulate. One type of influenza that has worried public health experts for decades is avian influenza, or bird flu, particularly the H5N1 strain. This virus primarily infects birds, but it can sometimes jump to humans, where it can cause a very severe and often fatal illness. The fear is that H5N1 could one day mutate to spread easily between people, sparking a deadly pandemic. To prepare for this threat, governments around the world have created stockpiles of H5N1 vaccines. These vaccines were developed years ago, based on the specific H5N1 viruses that were circulating at that time. For example, many stockpiles contain a vaccine based on a strain from Vietnam in 2004. The idea was that if a pandemic started, these vaccines could be deployed quickly to protect frontline workers and the public. It was a strategy of preparedness, an attempt to have a weapon ready before the war even started. This seemed like a sensible plan to get ahead of a potential global crisis. However, the problem with influenza is its constant evolution. A recent study showed that these stockpiled vaccines may no longer work. The H5N1 virus has not stood still since 2004. It has continued to mutate and change, a process known as antigenic drift. The H5N1 viruses currently circulating in animals, such as the one found in dairy cattle in the United States in 2024, are genetically very different from the older strains used to make the stockpiled vaccines. When scientists tested the old vaccine against the new virus in animal studies, it failed to provide protection. The immune response it generated was no longer a good match for the modern virus. This represents a different kind of vaccine failure. It is not a failure of the vaccine technology itself, nor is it a safety issue. It is a failure of matching the vaccine to its target. The virus is a moving target, and a vaccine designed for yesterday's virus may be useless against today's. This highlights a major challenge in pandemic preparedness. It is not enough to simply have a vaccine on a shelf. We need systems that can rapidly update and produce new vaccines as soon as a new threatening strain emerges. The lesson from the H5N1 stockpile is that our strategies must be as dynamic and adaptable as the viruses we are trying to fight. Section 7. The AstraZeneca COVID-19 vaccine. A complicated story. When the COVID-19 pandemic swept across the globe, the race to develop a vaccine was on. One of the leading contenders was the vaccine developed by the University of Oxford and the pharmaceutical company AstraZeneca. Unlike the mRNA vaccines from Pfizer and Moderna, this one used a different technology. It used a harmless, modified cold virus from chimpanzees to carry the genetic instructions for the coronavirus spike protein into our cells. This DNA-based vaccine was seen as a major hope for the world because it was cheaper to produce and easier to store than the mRNA vaccines, making it ideal for global distribution. The vaccine, known as Vaxzevria or Covishield, was rolled out in many countries in early 2021. It was a critical tool in the fight against the pandemic, saving countless lives by preventing severe illness and death. However, soon after its widespread use began, reports started to emerge from Europe about a very rare but serious side effect. A small number of people, mostly younger women, developed an unusual condition involving blood clots combined with low levels of platelets. This condition, called thrombosis with thrombocytopenia syndrome, or TTS, was very dangerous and in some cases fatal. This safety concern created a major problem. Even though the risk was extremely small, estimated at just a few cases per million doses, the severity of the condition caused widespread alarm. Several countries paused their use of the AstraZeneca vaccine or restricted its use to older populations, who appeared to be at a lower risk for this specific side effect. The vaccine was never approved for use in the United States. This situation was complicated. The vaccine was clearly effective in saving many lives, but the rare risk of TTS damaged public confidence and created regulatory challenges. Ultimately, the story of the AstraZeneca vaccine ended with its withdrawal from the market in 2024. The official reason given was that there was now a surplus of newer vaccines that were updated to target more recent variants of the virus. However, the withdrawal also came after the company acknowledged in court documents that its vaccine could, in very rare instances, cause TTS. This case is a complex example of vaccine failure. It was not a failure of effectiveness, but a failure of safety in a very small subset of people, which, combined with supply issues and the availability of alternatives, led to its eventual demise. Section 8. When society says no, the social failure of vaccines. Sometimes a vaccine can be a brilliant scientific success but still fail in the real world. This happens when people refuse to take it. This is not a scientific failure, it is a social failure. The reasons for this are often complex, rooted in history, politics, and personal beliefs. A perfect example is the resistance to the very first vaccine, the one for smallpox. Even in the 19th century, as soon as governments in places like Massachusetts tried to make the smallpox vaccine mandatory to protect public health, opposition groups emerged. They argued that the government did not have the right to force them to undergo a medical procedure. These early anti-vaccine movements were not based on a lack of scientific evidence. They were based on principles of personal liberty and a deep distrust of government authority. The first anti-vaccine society in the United States was formed in 1879, and it fought against these mandates in courts and in public opinion. While the Supreme Court in 1905 ultimately upheld the state's power to mandate vaccines for the good of the community, these groups were successful in pushing for exemptions. They argued for the right to conscientious objection, allowing people to opt out of vaccination based on their personal beliefs, a concept that continues to shape vaccine policy today. This historical tension has never gone away. In fact, in recent years, it has become much more pronounced. The debate over vaccine mandates and personal freedom has moved from the fringes of society into the center of mainstream political discourse. What was once a settled matter of public health has become a deeply polarizing issue. the COVID-19 pandemic amplified this divide to an unprecedented degree. Misinformation spread rapidly online, fueling distrust in scientists, public health institutions, and the vaccines themselves. Even highly effective vaccines failed to reach their full potential because a significant portion of the population chose not to get them. This modern resistance represents a serious failure for public health. A vaccine only works to its fullest potential when a large percentage of the population, known as herd immunity, is protected. When vaccine uptake is low, diseases that were once under control can make a comeback. We are seeing this now with outbreaks of measles in communities with low vaccination rates. The failure in this case is not in the syringe. It is a failure of communication, trust, and social cohesion. It shows that having a safe and effective vaccine is only half the battle. Convincing people to take it is the other equally important half. Section 9. Learning from failure, the path to better vaccines. In science, failure is not the end. It is often the beginning of a deeper understanding. Every vaccine that did not work as planned has provided invaluable lessons that have made all subsequent vaccines better and safer. The tragic Qatar incident in 1955, where live poliovirus was accidentally included in vaccine batches, was a horrific failure of manufacturing. But the response to that failure created the modern, rigorous regulatory system that oversees vaccine production today. It led to strict rules for testing every single batch of vaccine to ensure it is both safe and potent, a system that protects us all. The failure of the 1960s RSV vaccine was a humbling scientific lesson. It taught us that the immune system is far more complex than we imagined, and that a poorly designed vaccine can sometimes make a disease worse. This led to decades of research into immunology, helping scientists understand the difference between a good immune response and a bad one. This knowledge is now fundamental to how all new vaccines are designed. Scientists now carefully study the type of immune response a vaccine candidate generates to ensure it is protective and not harmful, a direct result of learning from that early, tragic RSV trial. The stories of RotaShield and the AstraZeneca vaccine highlight the paramount importance of post-market surveillance. Both vaccines were effective, but they caused very rare, serious side effects that were only detected after they were given to millions of people. The quick identification of these risks and the subsequent actions, withdrawing RotaShield and restricting AstraZeneca, demonstrate the strength of our safety monitoring systems. These failures reinforce the principle that the bar for safety must be incredibly high, especially for vaccines given to healthy people. They have led to even more robust systems for tracking adverse events in the real world. Ultimately, the history of vaccine failures has not only improved our technology, but has also reshaped our very definition of what a vaccine is and what it should do. We have moved from simple killed or weakened germs to sophisticated genetic platforms like mRNA and DNA. We understand that a vaccine must not only be effective, but must also be manufactured perfectly, induce the right kind of immunity, and have an impeccable safety profile. Failure is a harsh but effective teacher. Each mistake, each unexpected outcome has been a stepping stone on the path toward creating the safer, more effective vaccines we rely on today.